Hello everybody, welcome to your very own trusted channel Vigyan Ghar. Here we are with the third part of JNU 2021 MSc Life Science paper. We hope this will be helpful for your examination preparation. So let us start with the first question. The large cotyledon of grass family monocot plant is called. Now let's have a look at this particular figure here. In the grass family, the cotyledon is called scutellum. It is situated towards one side of the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cap, which is enclosed in an undifferentiated sheet called coleorhiza. So the correct answer to this question is option number B, scutellum. The next question is, an inflorescence is said to be determinate when only a finite number of flowers develop on meristem. Inflorescence meristem terminates into a flower. The production of new flower in axillary positions stop, or all flowers of inflorescence are of same sex. Now let's have a look at this figure here. The arrangement of flowers on floral axis is known as inflorescence. The inflorescence is classified into two main types: racemose inflorescence, which is indeterminate inflorescence, and cymose inflorescence, which is determinate inflorescence. Now in racemose inflorescence, the branch do not terminate into a flower. The flowers are arranged laterally in an acropetal manner. That is, the older flower are present on the lower side and younger flower are arranged on the upper side. In this type of inflorescence, the peduncle or the flower axis continues to grow. In cymose inflorescence, the peduncle end with the flower. Here the oldest flower are thus the terminal and the young buds are located in the lateral position. So from the given options now, statement B, inflorescence meristem terminates into a flower, is correct. Hence, in the options, option B only is the right answer. In the next question, read the following statements regarding cereal seeds. Statement 1 says the cereal seeds contain starchy endoplasm and eleuron cells. Statement B or 2 is at the maturity, starchy endoplasm dies but does not undergo autolysis. Statement 3. Eleuron cells remain alive till germination. Choose the correct statement. Cereal endosperms contain three major types of cells: starchy endosperm, transfer cells, and eleurons. Starchy endosperm cells are large, irregularly shaped cells and contain starch grains, while eleuron cells are cuboidal in shape and contain proteinaceous eleuron grains. Now, in starchy endosperm, cells undergo programmed cell death during the maturation phase of seed development, but these corpses are largely retained until germination. Eleuron cells instead remain alive throughout endosperm development, but they die and disassemble as a result of activation of a specific autolytic program during germination. Hydrolases released by dying eleuron cells help digest and remobilize the storage compounds in starchy endoplasm to support the seedling development. So, in the given statements here, all these statements are correct. So, our option number D is the right answer. The next question is, water uptake decreases when roots are subjected to A. Low temperature, B. Anaerobic conditions, C. Treated with respiratory inhibitors, and D. High temperature. Choose the most appropriate answer from the options given below. Among roots, water channel activity has been shown to exist, which may be inhibited by heavy metals, hypoxia, nutrient deficiency, low temperature, and high salinity. So the correct answer is option number B, statement A, B, and C only. Next question is, which of the following is or are produced during non-cyclic photophosphorylation? The photophosphorylation process, which results in the movement of electrons in a non-cyclic manner for synthesizing ATP molecules using the energy from the excited electrons that are provided by photosystem 2, is called as non-cyclic photophosphorylation. During non-cyclic photophosphorylation, the electrons released by P700 are carried by primary acceptors and finally passed on to NADP. Here, the electrons combine with protons, which is H positive, which is produced by splitting a water molecule and reduces NADP to NADPH2. The correct answer here is NADPH and ATP, that is option number C. The next question is, phosphoenol pyruvate is a major CO2 acceptor for which of the following plants? And the options are cowpea, apple, sugarcane, and pinus. The primary CO2 acceptor of C3 and C4 plants is RUBP, which is ribulose 1,5-bisphosphate, and PEP, phosphoenol pyruvate, respectively. Plants which do not have adaptation to combat photorespiration, a wasteful process, such plants are known as C3 plants. Now, C4 plants such as millets, maize, sugarcane, and tropical savanna grasses are adapted to stay in dry, hot conditions and can effectively combat photorespiration, can fix the carbon dioxide more efficiently than C3 plants. So here the correct answer is option number C, sugarcane. The next question is, which of the following is or are correct regarding the role of given phytohormones? A. Auxin stimulates fruit development. B. Ethylene promotes ripening of fruits. C. Cytokinin promotes senescence in leaf. And D. ABA inhibits stomatal closure. Actually, auxin is produced by seeds and it stimulates fruit development with gibberellin and delays the senescence. Cytokinin or CK is a phytohormone that promotes cell division and delays senescence in plants by improving nutrient mobilization. The ABA inhibits blue light dependent stomatal opening, an effect essential for promoting stomatal closure in the daytime to prevent water loss. Ethylene is highly effective in fruit ripening. So, from the given options, the correct answer is option number B, statement A and B only. The next question is, among the plant processes, which of the following are primarily regulated by phytochromes? A. Shade avoidance. B. Stomatal opening. C. Chloroplast movement. And D. Flowering. Phytochrome is a blue proteinaceous pigment that occur in two interconvertible forms. PR absorbs red light and PFR, which absorbs far red light, which is an active form of phytochrome. Now, photomodulation of flowering is phytochrome regulated process. In short days, PR form of the phytochrome promotes formation of flowering stimulus, whereas in long day plants, the flowering stimulus is promoted by PFR form of the phytochrome. Now, the shade avoidance response is also controlled by phytochrome photoreceptors. So, the low red to far red light ratios enhance ethylene production in wild-type tobacco, resulting in shade avoidance response, whereas ethylene insensitive plants show reduced shade avoidance response. The correct answer here is option number D, A and D statement, which is shade avoidance and flowering. The next question is match list 1 with list 2. So let's have a look at the list 1, Addison's disease. Now this Addison's disease is also known as adrenal insufficiency, which occurs in the body when body doesn't produce enough of adrenal hormones. So option number A matches with second, which is inadequate adrenal hormone. Acromegaly is a rare condition in which the resultant is excessive production of growth hormone by the pituitary gland, which causes enlarged face bones and bones in feet and hand. So here for option B, the right answer is oversecretion of growth hormone. Gout is a type of arthritis that causes inflammation of joint due to excess accumulation of uric acid. So option number C matches with 1. So let's have a look here. A matches with 2, B matches with 3, and C matches with 1. So the correct answer is option number D. The next question is, which of the following is the upper lip of cockroach? So now let's have a look at the mouth parts of cockroach. They include labrum, mandible, maxillae, and labium. Labrum or the upper lip is a broad, roughly rectangular shaped structure. It hangs from the front edge of the head to lower side. So the correct answer to this question is option number A, labrum. The next question is match list 1 with list 2. So here let's have a look at the options in list 1. Silverfish. Silverfish is a species of small primitive wingless insect. Its name comes from its look. So it looks like a silvery light gray fish-like appearance it gives. So it is called a silverfish, but it's actually an insect. So it matches with 3. Obelia is a marine colonial trimorphic hydrozoan which possesses polyp, medusa, and blastocyst stage in its life history. So option number B, medusa, matches with Obelia. Option number C, spicules. Basically, spicules are calcareous structures that form a supporting skeleton in sponges. So option number C, spicule, matches with sponges, that is 4. And a cetacean is a genus Delphinus and elite species Cetacea is an infraorder that comprises of 94 species of whales and dolphins. So here the option number D, dolphin, matches with second cetacean. So the correct answer is A matches with 3, B matches with 1, C matches with 4 and D matches with 2. So the answer is option number B. Next question is, which of the following statement is not correct for aquatic annelids such as Nereis? It has lateral appendages which help in swimming. It has closed circulatory system. It has neural system containing paired ganglions, and reproduction occurs through both sexual as well as asexual mode. The correct answer to this question is option number D. Reproduction occurs through both sexual as well as asexual mode. The reproduction process of Nereis is quite simple. The sexes are separated in Nereis, and when the organism is sexually mature, the body transforms into epitoke. When transformation occurs, the body contains gametes for reproduction. Now, the gonads are temporary structure and appear only in the breeding season. Here, the ovary and testes degenerate after the liberation of sex cells into the outside water. So, the fertilization that occurs is external and occurs in seawater. So Nereis only reproduces via sexual mode rather than asexual mode. So the correct answer is option D. Next question is, given below are two statements. Statement 1. When mRNA of aquaporin protein is inserted into frog oocyte, aquaporin is expressed and it is incorporated in the membrane of oocyte. Such aquaporin expressing oocyte swelled when placed in water. Statement 2. When mRNA of aquaporin protein is inserted into frog oocyte, aquaporin is expressed and it is incorporated in the membrane of oocyte. Such aquaporin expressing oocyte shrunk and died when placed in water. In the light of the above statement, choose the correct answer from the options given below. So when the cell membrane of a variety of different bacteria, fungi, animals and plant cells contain aquaporin, through which water can flow more rapidly in and out of the cell than by diffusing through the phospholipid bilayer. When Peter Agre and colleagues compared frog oocyte expressing aquaporin with control oocytes that lack aquaporin and they were placed in water, they found that oocyte expressing aquaporin swelled and burst up as the water was transported into the cell. So here statement 1 is correct and statement 2 is false, and our right answer is option number C. The next question is, in case of Drosophila, after the cells are formed from syncytial embryo, there are cell-to-cell interaction mediated by segment polarity gene. In this aspect, the following statements were given. A. Segment polarity gene reinforces the parasegmental periodicity established by earlier transcription factors. Though these cell-to-cell interaction cell fate are established within each parasegment, through these cell-to-cell signaling, cell fates are established within each segment. Segment polarity genes encode proteins that are constituent of winged and hedgehog signaling pathway. And Fujita-Raju and Hairy are examples of segment polarity gene. So now let's have a look here. The statement A, B and D are correct. It is the function of the segment polarity gene to reinforce the polarity of parasegments as established by the pair rule genes. Now the cell fate is established within each parasegment, that is statement B, not the segments. Segment polarity genes include hedgehog, engrailed, wingless, etc. And for the statement E, it is wrong because Fujita-Raju and Hairy are examples of pair rule genes and not segment polarity genes. So here our right answer is option number C. The next question is, movement of individual cells into blastocele during gastrulation is called ingression, involution, invagination or delamination. So here we know that there are two general types of morphogenic movements that can be recognized. Either individual cells, they move by migration or ingression, and the groups of cells that move by invagination, involution, epibole, delamination or convergent extension. Here, for this question, the correct answer is option number A, ingression, which is the movement of individual cells or small groups from epithelium into the cavity. Next question is, given below are two statements. One is labeled as assertion and other is labeled as reason. Assertion: tendril of pea and leaf-like organ of cactus are homologous organs. Reason: homologous organs have similar evolutionary origin. In the light of the above statements, choose the most appropriate answer from the options given below. Homologous organs have similar origin, but due to evolution, the organisms undergo developmental changes in order to adjust in the changing environmental condition. In many plant species, leaves have been modified for functions other than photosynthesis. The cactus plant, like Opuntia, grow in desert area. In order to prevent the loss of water in such arid condition, the leaves have been modified into spines. The flattened stem-like structure called phylloclade has been modified into fleshy succulent flat green structure which can perform photosynthesis and prepare the food similar to leaves. The cactus spines, which is not the leaf like phylloclade, appear tendril although quite different in appearance are homologous because both of them are modified leaves but with different functions. So here the correct answer is A is not correct but R is correct. That is option number D. The next question is pneumatophores are mostly present in. Options are mesophyte, helophyte, xerophyte and hydrophyte. A number of helophyte plants possess small negatively geotrophic vertical roots called as pneumatophores. These special types of breathing roots stemmed out from the subterranean root system have lenticels for gaseous exchange. So the correct answer here is option number B, helophytes. The next question is, in some small fishes that gather around and feed on the leftover of bigger animals like shark. This relationship is an example of mutualism, parasitism, commensalism, or predatorism. Commensalism is an association between two organisms in which one benefits and other derives neither benefit nor harm. For example, as given in the question, the fishes gather around and eat scrape of prey that are dropped by the shark. So here the correct answer is option number C, commensalism. The next question is, which element is most abundant in our universe? Hydrogen is the most abundant element in the universe, which accounts for almost 73 to 75% of all matter in the universe. Helium is the second most abundant element with approximately 24%. So the correct answer here is option number A. The next question is, real-time PCR can be used for detecting the viral load in animal tissue, bacterial load in plant tissue, amount of viral cap protein present in any tissue, and amount of bacterial cell wall component in any tissue. The use of real-time polymerase reaction in the molecular diagnostic has increased to the point where it is now accepted as a gold standard for detecting the presence of specific genetic material in any pathogen, including a bacteria or virus. Real-time detection of PCR product is enabled by inclusion of a sequence-specific primer of fluorescent reporter molecule in each reaction, that yields increased fluorescence as well as increased amount of product cDNA. So the correct answer in the options is option number B, A, viral load in animal tissue, and B, bacterial load in plant tissue. The next question is, which of the following methods can help us estimate the molecular weight of a protein? Options are gel electrophoresis, gel exclusion chromatography, bioinformatic analysis of amino acid sequence, and spectrophotometry. In a protein, once it is isolated, the most common method to estimate its molecular weight is by running it on a protein gel, which is known as the SDS page. A marker enzyme is run along with it to know the molecular weight of the protein. Size exclusion chromatography or gel filtration is also a widely used method to determine the molecular weight of native protein. The molecular weight of a protein can also be determined by knowing its sequence of amino acids. For any given protein, for example, we can roughly estimate the molecular weight of protein by multiplying it with the average molecular weight of amino acids, that is 110 Dalton, with the total number of amino acids present in the protein. Thus, for example, if we need to calculate the molecular weight of a nonapeptide hormone, oxytocin, all we need to do is multiply 110 with 9 to get an approximate molecular weight of the hormone. The correct answer here is option number C, which refers to statement A, B, and C only. The next question is, ninhydrin reagent is primarily used for detecting. Ninhydrin is a chemical test that is used to check whether a given annihilate contains amines or alpha amino acid. In this test, ninhydrin is added to test solution of the annihilate. The development of deep blue color indicates the presence of ammonia, primary or secondary amines, or amino acids with free amino group in the annihilate. So the correct answer here is option number D, ammonia and amines. The next question is, which of the following is or are DNA binding dyes? Now here we have options DAPI, Nile red, ethidium bromide and trypan blue. Ethidium bromide is likely the most well-known dye which is used to visualize DNA. Molecules of dye adhere with the DNA strand and fluoresce under UV light, showing you exactly where the bands are present in the gel. Then we also use DAPI, which is another fluorescent stain that can bind strongly to AT-rich regions in the DNA. And along with it, Eva green is also one fluorescent dye that has been used to inhibit PCR to a lesser extent than any other dye. That makes it useful for use in quantitative PCR. Nile red is a phenol-xenon lipophilic dye that shows strong fluorescence enhancement upon transition from aqueous to hydrophobic environment, such as the protein SDS complexes. Trypan blue, on the other hand, is a vital stain which is used to identify dead tissue from the alive tissue. So, the alive tissue do not allow the trypan blue to enter into the membrane, while the dead tissue, because the membrane is permeable, so it allows the trypan blue to enter into the cell, and hence the dead cell appear distinctively blue under microscope. So from the given options, option number A and C only, that is DAPI and ethidium bromide, can be used as DNA binding dyes. Next question is, which of the following is a quorum sensing molecule for bacteria? Quorum sensing is a method of communication between bacteria that enables the coordination of group-based behaviors based on population density. They signal change in gene expression, such as switching between flagella gene and gene of phyli for development of biofilm. N-acyl homoserine lactones are a class of important signaling molecules produced by acyl homoserine lactone synthase involved in quorum sensing process found in numerous gram-negative bacterial species. So here the right answer is option number A, N-acyl homoserine lactones. This brings us to the end of this video. Please do not forget to like and share this video with your other friends, and to subscribe to our channel for more such content. Bye bye.